Hi guys, Mrs. Homan here with another science review video. This one is on meiosis. So the first thing I'm going to talk about is the difference between meiosis and mitosis. So right here in this first circle, this is going to be our parent cell. That parent cell for both of these has 46. This is because we're talking about genetics and heredity. We're really talking about humans right now. And humans have 46 chromosomes in all of our body cells or somatic cells. So all different species have all different numbers. Again, I'm just going to focus on humans right now. So in this original parent cell, we have 46 chromosomes or chromatids. During interphase, that occurs before both meiosis and mitosis occur, DNA is copied. So we go from 46 chromatids to 92 chromatids. Okay? So in mitosis, we go from our original parent cell, we duplicate that DNA, mitosis occurs, and we end up with two genetically identical cells. Now, these are both genetically identical to each other and to that original cell, okay? So these two are identical and these are identical, okay? So remember, mitosis is, its purpose is growth and repair and asexual reproduction. Okay? And so mitosis ends up from one parent cell, two cells are created that are genetically identical. It is used for growth, repair, and asexual reproduction. Meiosis, however, we have our parent cell, 46 chromatids. It gets duplicated to 92 chromatids. During prophase of meiosis 1, which I'm going to talk a little bit more about in a few minutes, crossing over or recombination occurs. So it mixes up the DNA between those chromatids so that at the end of meiosis 1, while we do end up with the same number of chromatids as that parent cell, and it looks very similar to what happened here, these are no longer genetically identical to that original cell. And therefore, meiosis 2 occurs. These are split up again. We get 23 chromatids in each one of these 4 daughter cells. And these are genetically different from each other and from that original cell. So not one of these is the same, and they are not the same as that original cell. So for starters, we can tell that already because this one has 46 chromosomes, this one has 23. But additionally, after recombination, all of these will be different. So the result of meiosis is 4 genetically different cells, and it is used only for sexual reproduction. Okay? Gametes are the only cells in our body that undergo meiosis. Every other cell, eye cells, lung cells, heart cells, they all go through mitosis. Again, meiosis is the only occurs in the gonads to create gametes. Mitosis creates those somatic cells. So now let's dive deeper into interphase. So interphase, that phase that occurs before both mitosis and meiosis occur, DNA is unwound in the form of chromatin. So we see we have All this DNA is all unwound. It's being replicated. And again, this occurs before both mitosis and meiosis. So before I really begin going through the steps of meiosis, I want to talk about what's occurring right here during interphase. So what's happening inside the nucleus during interphase? So prior to interphase, so for simplicity's sake, I'm using a cell with only 4 chromosomes. Okay, so 1 from mom, 1 from dad, 1 from mom, 1 from dad. And these would be homologous chromosomes, and these would be homologous chromosomes. So during interphase, that DNA is copied. So we began with 4 chromosomes. After interphase, we have 8 chromatids. Still 4 chromosomes because they're connected right there at the centromere, but 8 chromatids. 1, 2, 3, 4, 5, 6, 7, 8. So this is why interphase has to occur first. Interphase duplicates that genetic material so that we will end up with the correct number of chromosomes or chromatids in those final cells. So prophase 1 is the first step of meiosis 1. Now, arguably the most important part of meiosis is this process of crossing over, also called recombination, that occurs during prophase 1 of meiosis only. So these homologous chromosomes pair up to form a tetrad. So tetrad meaning 4, 1, 2, 3, 4 of those chromatids pair up, and they quite literally cross over each other. They stretch their little arms over each other and they exchange genetic material. And I hope you can see this well enough. But here I've drawn the result where we end up with 4 genetically different chromatids. This one has mostly red, a little bit of orange, mostly red, a little bit of orange, but different than this mostly red, little bit of orange. This one, mostly orange, little bit of red, mostly orange, little bit of red. Again, but different than this mostly orange and little bit of red. And this is how we end up with this right here. These 4 genetically different cells. After prophase 1 has occurred, after that crossing over has occurred, none of these chromatids are exactly like the original. So now the original was all red and all orange, but our result is all 4 chromatids are unlike each other and unlike the original. Now, for simplicity's sake, in my model, in my diagram I'm about to show you, I've just exchanged 1 whole arm just because it would be kind of hard for me to draw this over and over and you guys to really be able to see what's happening. But I wanted to show you this so that we really understand where this genetic diversity is coming from, where the genetic variation is coming from, how we get 4 different cells, okay? So let's start. We're going to start up here. So prophase 1, this is kind of what it would look like. We have each chromosome pairs up with its corresponding homologous chromosome to form a tetrad. Recombination and crossing over is occurring here, and this nuclear membrane is breaking down. The spindle fibers are moving across between these 2 centrioles, and those 2 centrioles are moving to opposite ends of the cell. Now, here we can really see where I have exchanged that genetic material, where I have 1 red arm on this mostly orange chromosome and 1 orange chromosome, one orange arm on this mostly red chromosome, and the same with these blue and green ones. So metaphase 1, these guys are lining up along the center of the cell. This is the metaphase plate. It is an imaginary line along the center of that cell. Those spindle fibers are going to extend from the centrioles, attaching to the centromere or center of each chromosome. Our next phase is anaphase. You can remember that by A for away. I have my imaginary bright pink arrows here to show that these are moving away from the center of the cell towards opposite ends of the cell. And then the last part of meiosis 1 is telophase 1 and cytokinesis. So the nuclear membranes start to reform, and that cell divides the cytoplasm into 2 separate cells. Now notice, we've already got different than what we started with. We started with 1 each of red, orange, blue, and green. And here, we do not have that in either of these, even though each has 4 chromatids like that original cell. Okay, but mostly red, only a little bit of orange, mostly blue, only a little bit of green. Orange, red and orange, green, blue and green. So not full red, full orange, full blue, full green. Okay, so now we're going to move on to prophase 2. So this is starting meiosis 2. These nuclear membranes are starting to break down again. Those centrioles and spindle fibers are moving to opposite ends of the cell. And the most important thing is that the chromosomes are not copied again, and recombination / crossing over does not happen again. Okay? Metaphase 2, they're lining up along the center of the cell, but unlike in metaphase 1, they're not pairing up on either side of that metaphase plate. They're lining up directly along that metaphase plate. So the way to be able to tell if we're in metaphase 1 or metaphase 2 is if they're paired up along the center or in a single file line along the center. So those chromosomes line up, the spindle fibers extend and attach to those centrioles of the cell. And now in anaphase 2, these guys are getting split apart. They're getting ripped up into individual chromatids. So we have this one is all red moving to this side, all blue moving to this side. This one is orange and red, green and blue moving to this side. And the same is happening over here. Again, A for away, they're just separating. And then last, we have telophase 2 and cytokinesis. That nuclear membrane is reforming. The cell divides the cytoplasm into 2 cells. So now we have 4 total, so each divided into 2. And each cell has half the usual number of chromosomes. So I want us to see this. Each one of these now has 2. We began each with 4. So now we have half the genetic material, and not one of these is exactly the same as each other or exactly the same as the original. So here we have 1 red, 1 blue, half orange, half red, half green, half blue, but different than this one. So this had a top orange, this one has a red orange, so these ones are switched, so these two are not the same, and then this one is orange and green. So not one of these 4 are like each other, and not one of those 4 is like the original. Okay? So I hope this was helpful, and have a great day.